On a personal note, I kept bees way before varroa arrived. Where we were all encouraged at the time to treat with hard chemicals. Then, of course, after about 8 or 10 years, resistance came. So it's taken us nearly 30 years to take resistance seriously, and some beekeepers, I'm afraid, still haven't. Now, if Tropilaelaps arrives, we could make the same mistakes again. And having had the experience, I certainly don't recommend that course. Along with small hive beetle, Tropilaelaps was made notifiable in 2006. So 19 years ago, it was considered that serious by the government. We've actually done very little until Maggie's team started working on it recently. And I will draw your attention to a video of Maggie's colleague Dan Etheridge on the National Honey Show YouTube channel. This webinar will give the latest information, hopefully. Now, it will be recorded, but if you are viewing the recording, please remember that because there are new discoveries really all the time, the content may be superseded or augmented. Now, what we'll try to do is keep beekeepers up to date on the BIBBA website, where there's already some information that's been written by Maggie and placed on there. Please, please, please ask your local beekeeping associations to put a link to it, and also this video and Dan's videos as well. So that's all from me. So if you're in the starting blocks, Maggie, off you go now. And
okay. I will just share my screen. So hopefully you should be able to see that now. Is that all working okay for everybody?
Yep, that's there.
Got that, yeah.
All right, brilliant. Okay, and everybody can hear me clearly enough?
Yep.
Right, okay. Well, yeah, good evening, everybody. Thank you for having me and turning out to listen to my talk. I'm going to give a bit of an overview of, what Tropilaelaps is first, and then sort of go into a few bits of the latest research, because our understanding of this mite is kind of evolving and developing. So first things first, what are Tropilaelaps mites? And they are a parasitic mite, and they're similar to varroa in that they infest and they reproduce and they feed on honeybee brood. So they are indigenous to Asia, in the same way that varroa was, and these mites naturally infest the giant Asian honeybees, Apis dorsata, and that's what you can see here in this video and the photographs there. So in the UK, we have the Western honeybee, mellifera. That's the only honeybee species that we have. But in Asia, there are multiple species of honeybee, and the giant Asian honeybee is just one of those. So these bees, as you can see, they're quite a bit bigger than our native honeybees. They nest out in the open. They build this large single comb, and it has honey at the top where it attaches to the branch. And then the brood is sort of built underneath, and the bees hang on the comb like that to protect the brood and to protect the colony. And they'll build these nests sort of up in tall trees and high up on the side buildings. So here you can see there's a very large tree there, and I've put in a picture of me just to kind of give you an idea of the scale of these trees. And into the side of that tree, I think I've got a little pointer here, if I can get that to work. So there are bamboo spikes that are driven into the side of the tree that go all the way up. And these spikes are used as ladders by local honey hunters to access these colonies. And generally, a honey hunter will only take a small portion of the honey or the brood because they don't want to harm the colonies. The picture of the comb there that's in the middle picture was actually that the whole comb was collected. It gives you a bit of an idea of size. This was part of a research project, so that doesn't normally happen. So in this tree, you can see that there are several colonies. So you've got the colonies here along this branch. Hopefully you'll be able to see sort of along this branch, there's other colonies that nest there. You've got a colony there on that branch. And then again on this branch here. And the interesting thing is all of these bees will be related to one another. So you've got sort of mother and daughter colonies, sister colonies, and they'll all nest in the same tree. And they'll stay in a location like this for perhaps sort of 3 or 4 months. And they'll utilize the nectar source and the pollen source that's in that area. And when that sort of starts to run out, they actually migrate. Now, Tropilaelaps and the giant Asian honeybees, they have co-evolved, and Apis dorsata has developed strategies to deal with Tropilaelaps, such as migrating. So when the colony migrates, they leave the combs behind. They leave behind any brood that hasn't hatched out. This reduces the amount of Tropilaelaps mites in the colony. And Apis dorsata will spend several weeks, sometimes even months, migrating across the country to find a location that has a good source of nectar and pollen. And during this time, they do what's called bivouacking. So they will form what we would probably call like a swarm cluster. So if you see a swarm that's sort of hung up in a shrub or a tree or something, and it's a cluster of bees. And Apis dorsata do that, and they still feed each other, and they'll go out and forage. But they will have several weeks, sometimes even months, where they don't build any combs and they don't have any brood. And the interesting thing is that Tropilaelaps survive during this migration when there isn't any brood, and nobody really sort of understands how they survive. But with any parasite-host relationship, Tropilaelaps doesn't generally cause Apis dorsata, so these giant Asian honeybees, too many issues, because a parasite, it doesn't want to kill its host. So unfortunately, though, Tropilaelaps has jumped species to our Western honeybees. So, we took Western honeybees to Asia. It probably started around the 1890s in Thailand, which is where I've done most of my work. Western honeybees weren't introduced until the 1950s, but all through the early 1900s, we were transporting Western honeybees to Asia because they're easier to manage, they're more productive than the native species of honeybee. So if you had the choice of, say, climbing that dirty great tree and you'd collect maybe 20 pounds of honey versus you've got a nice box of bees in your garden that's easy to access, you can see why Western honeybees were introduced and why they became popular there. The problem, though, is that Western honeybees, Apis mellifera, are closely related to Apis dorsata. So they are both Apis species of bee and both honeybees, which means that they are similar enough for parasites to jump species and for these parasites to be able to survive and reproduce on them. So the same thing happened with varroa. Varroa mites naturally come from the Asian honeybee, Apis cerana. Varroa did the same thing, jumped species to our Western honeybee, and that's how Varroa eventually ended up in the UK. So here we've got a video of some varroa mites and some Tropilaelaps on honeybee larvae. The Tropilaelaps are smaller, so they're smaller than the varroa. They're about 1 millimeter long by about 0.5 millimeter wide. And just in this video here, once it sort of circles around again, I'll point out some things. But there are 4 subspecies of Tropilaelaps, and only 2 of those are harmful to our Western honeybee, which is Tropilaelaps mercedesi, which is what we have here, and Tropilaelaps clari. So they can reproduce more quickly than varroa, so I'll go into that in a second. So what I wanted to show you here was, this one's a little bit damaged, so that one's not as quick as the other ones. This one, when she comes back, is a pregnant female. You'll see that she's fatter than her sister here. And we witness this behavior where they will harass the varroa mites. So they reproduce more quickly than varroa do. They also cause more damage to the brood when they feed. They vector different viruses, and they're very hard to detect in a colony because the symptoms and the damage that they cause is very similar to that varroa causes. A bit of a comparison of the 2 different life cycles here. So with varroa, you have your foundress female mite. She will go into a cell before it's sealed, and she'll hide in the brood food underneath the larvae. And when that cell is sealed, she'll come out and she'll lay her first egg. That first egg always hatches out to be a male mite, and she'll then lay subsequent eggs. And these mites that hatch out will be female varroa mites, and they all go through a series of molts before they are sort of fully formed and like viable varroa mites. and then the male mite will mate with its sisters. And when, depending on whether the original female went into a worker cell or a drone cell, on average, you will get 1.6 viable varroa mites from a worker cell and 2.6 from a drone cell. But like I said, Tropilaelaps, they can reproduce more quickly, so they can mate outside of the cells. The females can reproduce by parthenogenesis. So this means that even if the female hasn't mated, she can produce reproductive offspring, so reproductive male and female offspring, even if she hasn't mated. And just because of the rate of reproduction and the way that they molt, so they molt quicker than a varroa mite does, with an infested worker cell, on average, that will produce 3 viable Tropilaelaps mites, and from a drone, that increases to 4.5. So that's roughly double the rate of reproduction to that of varroa. So what would you expect to see in a colony infested with Tropilaelaps? There's a little video here of of a comb where the bees have been shaken off it. The mites are really difficult to see. They're a lot smaller. They're faster, and most beekeepers wouldn't routinely go around shaking the bees off their combs like this. So if you try to imagine seeing mites in a colony when you were just doing your normal everyday inspection with lots of bees on the frame, it's very difficult to see the phoretic mites in a colony. So you probably won't see the phoretic mites, but you might see symptoms of the damage they cause, and that's parasitic mite syndrome. And unfortunately, it's the same damage that varroa causes. So perforated cell cappings, you've got that patchy pepper pot brood pattern. They'll be dead, chewed down, cannibalized larvae and pupae, neglected brood. And very often with the adult bees, you'll see the deformed wings and the shortened abdomens, and just generally the colony gets weaker. The big difference that we've seen and we've heard anecdotal reports of is that Tropilaelaps will kill a colony much more quickly than varroa does. So in the space of about 2 to 3 months from the start of an infestation, Tropilaelaps can kill a colony. With varroa, if you don't treat, generally the colony, if the colony doesn't have any sort of natural resistance to it, it can take about sort of 3 years before that colony will succumb to a varroa infestation. So one of the reasons that Tropilaelaps cause such high colony mortality is because of the way they feed. So with varroa mites, you have that foundress female mite, and one of the first things she does is she'll set up a feeding site on the larvae, and all of the mites in that cell will communally feed from that one feeding site. Tropilaelaps, though, so they don't communally feed, and they will create multiple feeding sites. They also tear the brood when they feed. Varroa have piercing mouth parts. They're a much sort of neater and cleaner feeder than Tropilaelaps are. So there's a piece of research here where they looked at Tropilaelaps feeding, and they've stained these wounds that the Tropilaelaps mites cause. They've stained them with blue dye, and you can see that there's lots of wounds on the brood that these mites have caused. And the issue is that when that brood goes on to pupate and goes on to emerge as an adult bee, those wounds and that damage turns into sort of deformities in the adult bees. So you can see that these adult bees had deformities on their antennae and on their proboscis and on their feet and things like that. And all of these wounds and damage will shorten the life of the adult bee, but it also means that the adult bee is going to be less productive for the colony, which is one of the key reasons why the colony will succumb so quickly to a Tropilaelaps infestation. So I've included a QR code here that will take you to that bit of research. I kind of strongly recommend, if you're interested, go and have a look at it. It's a really good piece of work. So the other thing that Tropilaelaps do is they also vector viruses. So varroa mites do that when they feed. But these viruses are different strains of virus to the ones that our bees probably already have a bit of resistance to. So I kind of liken it to the difference between seasonal flu, where the UK population has a sort of general level of resistance to seasonal flu. It doesn't generally cause too many problems within the population, But then something like COVID-19 comes along, which is a different strain of a similar virus, and that has a devastating effect. So if you liken that to deformed wing virus in the bee population, the Tropilaelaps mites are sort of spreading these slightly different strains of the virus, which our bees don't have a resistance to. So that also causes a high level of colony losses. So in terms of distribution, we've got a map here. Shows the distribution of, so we've got the giant Asian honeybees, Apis dorsata, in the dark blue, and that's their natural range where you'd naturally find them in Southeast Asia. And the red areas are where Tropilaelaps mercedes have been confirmed, and Tropilaelaps mercedes is one of the species that is troublesome to our bees. And you can see that they've spread well beyond the range of the giant Asian honeybees. And they've done this on the other honeybee species like the Asian honeybees, but also beekeepers have spread them. So migratory beekeeping, queen cells, things like that. The red areas are where Tropilaelaps, so we've got these two areas where Tropilaelaps was confirmed in 2024. And in the spring, it was confirmed in the Krasnodar region of Russia. And then later on in 2024, also confirmed in Georgia for the first time. So this is the first confirmed report of Tropilaelaps in Europe. And beekeepers were anecdotally reporting Tropilaelaps in these areas since probably 2020, 2021. And it's taken sort of 3 to 4 years for that kind of anecdotal reports to actually be sort of scientifically confirmed and then for action to take place. So how else might Tropilaelaps get spread around the world? So I've mentioned beekeepers and migratory beekeeping and bee sales. One issue, so a few things happened in 2022 when I was National Bee Unit, which were sort of slightly concerning for us, and this kind of instigated the work that we did into Tropilaelaps. So first thing was in 2022 National Bee Unit, we were notified of so Apis florea. They are the red dwarf honeybee. They are another Asian species of honeybee that comes from Southeast Asia. And a colony came into Felixstowe docks on the side of a shipping container. That shipping container had come from India. Now, the workers at the docks there, they noticed this, and they reported it to the National Bee Unit and also to a pest controller. They actually thought that they were flies when they initially reported them, but the pest controller went out. He destroyed the colony, realised they were bees, and it was reported. But potentially that was a transmission route into the UK, because Apis florea, the red dwarf honeybees, they are an Apis species of honeybee. Tropilaelaps can breed and reproduce in their brood and on the bees. So this doesn't just happen in the UK. The photo on the left-hand side there, again, red dwarf honeybees. This is the actual photo of a colony on a shipping container that turned up in Western Australia. And this one had a queen and brood present. And Japan has also, they seem to be very good at finding these colonies on shipping containers. So they've had the giant Asian honeybees, they've had Apis cerana on boats, they've had Apis florea, and they find them and they intercept them. But it is concerning because it is a way that these mites can be spread around the world. The article on the right-hand side was in the Guardian newspaper just before Christmas. And this is the first report of Apis florea, so the red dwarf honeybees, and they've now been detected in Malta in Europe. The interesting part about this was that they weren't actually found at a port or an airport, but they were found inland. So this kind of suggests that they might have been there for a while. Potentially they might be established. Again, I've put the QR code in for that bit of work there, so you can go and read more about it and how they found it. But if a colony of bees, and it doesn't matter whether it's our Western honeybees or these dwarf honeybees, if they can build some comb and store some honey, whether it's a boat or a shipping container or an aeroplane, then potentially they're going to be able to survive, at sea for long enough to reach another country and potentially spread all sorts of things. It doesn't necessarily have to be Tropilaelaps, and there's other viruses and diseases as well as mites. So this is a piece of research which we just published at the end of last year. And we were interested to find out, what Tropilaelaps mercedes could survive on. And there's all sorts of different, the understanding about how they can survive is kind of changing and evolving, and this is something that we wanted to look at. And we were concerned about transmission, so we wanted to know, like, could Tropilaelaps survive on things, such as honeycomb, if that was imported into the UK, or such as pollen, for long enough to be able, could they survive for long enough to be able to reach us? So in 2023, we were in Thailand and we did a preliminary study, and we came up with ideas and we kind of brainstormed different scenarios where these mites might be able to reach the UK. So one example of those scenarios was that if we were in Thailand and we're working on the beehives and we had Tropilaelaps on our bee suits, and then we packed those up and we flew back to the UK, and then we've been away for several weeks. We would look at our own bees.
Maggie, you're breaking up a bit.
Potentially have in the UK.
Maggie.
And like I said, things. Sorry. Can you hear? Shall I?
Maggie, you broke up.
Can you hear me now, Roger? Is that? Or am I breaking up?
Yeah, carry on.
Okay,
sorry.
I'll carry on. Oh, no worries. All right, just shout again if I break up. Right, so this was the piece of work where we wanted to look at transmission and what could Tropilaelaps survive on. So in 2023, we did this preliminary study, and we wanted to look at things like honeycomb, dried pollen. So you can buy dried pollen in health food shops as a food supplement. A lot of that is produced in Asia and then shipped over to the UK. We also, we looked at the fabric and the bee suit cloth. So could we, as researchers, if we had Tropilaelaps on our clothes, could we bring them into the UK? Things like honey and that. And we also looked at things like dead bees and dead brood, and we ran this experiment. And at that point, we did a lot of reading, and the scientific literature said Tropilaelaps mites cannot survive for longer than 72 hours on anything other than honey larvae. And we'd given ourselves about 4 or 5 days to run this experiment. And the good news was that the Tropilaelaps mites, so they didn't survive for longer than 48 hours on honeycomb or on the dried pollen or on the fabric. But as I said, we'd also tried other things like these dead bees and dead brood. And it got to the point where, so the mites survived for longer than 72 hours. And then it got to the point where our time in Thailand had run out, and we were on sort of day 5 and day 6, and these mites still hadn't died, and we had to pack up. And this was sort of slightly concerning to us, because this kind of length of survival hadn't been reported before. And we had to come back to the UK. We had flights booked and things like that. So when we returned last year, we were keen to read the experiment, and we gave ourselves some more time. And we'd already ruled out things like the pollen and the honey and the fabric. So we kind of discarded those matrices, and we wanted to look at different things. So this time around in 2024, we looked at freshly emerged Apis mellifera adults, so Western honeybees. When those bees emerge out of the comb and they're all sort of young and cute and fluffy, we use some of those. We also used Apis dorsata, so the giant Asian honeybee adults. I sort of dangled out of a window at Chiang Mai University, and I collected some of those adults from the wild colonies. They're they're the natural hosts, and we thought, well, it's the natural host of the mite. The mite should be able to survive for quite a long time on them, without any brood. Again, we also did dead bees. We looked at dead pupae as well, because that's what we did in the previous experiment. And throughout the course of the experiment, everything was sort of kept in an incubator, and we kept all of the conditions the same. And the dead bees and the dead pupae that we were using, once they'd been in an incubator for a week, they were pretty revolting. They decomposed. There was sort of gloopy mess in the bottom of the pots that we used. And these are the results from that experiment. So there's a few interesting things which we were quite surprised. So firstly, Tropilaelaps on those giant Asian honeybees, its natural hosts, they didn't survive for longer than 48 hours on their hosts. And when we actually introduced the mites onto the bees got very agitated. They groomed themselves a lot. And yeah, like I said, they died relatively quickly. We didn't observe this, but a similar piece of research from back in the 1980s, and when they looked at the mites that they put on these Apis dorsata, the bees had actually done things like they'd bitten off their legs and they damaged their bodies. And certainly we saw the adult bees grooming themselves a lot. But the difference was with Apis mellifera, so our western honeybees, a large portion of the mites died quite quickly, so that was about 85%, but there was this small percentage that managed to survive for 120 hours, so up to 5 days. And what we suspect is happening is that if the mites are able to set up a feeding site on those newly emerged bees, then they can survive for quite a long time. But if they can't set up a feeding site, then they die, which is why only a small percentage would go on to survive. Another theory which some other researchers have, they haven't sort of investigated fully, but they seem to think that in the same way that Browler do, where Browler are able to get food from the bees via Tropilaelaps, that the Tropilaelaps mites are able to do that and are able to feed from the bee that way. The concerning result for this was the dead bees and the dead pupae. We saw really quite long survival for Tropilaelaps. So we had them surviving for up to 7 days and up to 8 days on these different matrices. And the concern here is that the mites can survive in things like old combs, old beekeeping equipment that hasn't been sterilised, and they could be transmitted to the UK in this sort of used beekeeping equipment, or like I said, sort of wild colonies on the side of shipping containers. If that colony dies, but there's still dead bees and dead brood available, the Tropilaelaps mites will be able to survive. And another possible scenario, which when you have things like queen imports and packaged bees, very often, not all of the bees will survive that. You will still have some attendant bees that have died in, say, that queen import. And while the mites were only surviving for a few days on the live adult bees, if the bees are dead, they can survive for almost double the length of time. And we get a lot of queen imports into the UK. Very often beekeepers will buy queens. They don't necessarily appreciate where these queens are produced. So this is quite a concerning thing. This is something that we're quite concerned about in terms of the transition into the UK. So another sort of preconception about Tropilaelaps is that they need brood to be able to survive. There's this perception that colonies will be broodless when it's cold in the winter, particularly in the UK, and beekeepers will treat for varroa with oxalic acid, and they believe that it's the middle of winter, they believe that their colonies are broodless. But if you actually go into the colonies and you start taking them apart, very often there will be some brood present. And this isn't just in the UK. There's been work by beekeepers in North America where it's very cold, Canada, places like that, where it's well below freezing, and they do see brood in their colonies. Now, our winters really aren't that cold, and particularly when you compare the UK to countries like Korea. So it's not unusual. I've put two kind of graphs here. So it's not unusual for Korea to have temperatures where it's below freezing for several months at a time. And by comparison, Scotland is quite considerably warmer. So Tropilaelaps mites have been established in South Korea since probably about 1995 to 2000, so approximately 20 years. And if they can survive in countries like South Korea, where they do have these harsher winds and they survive and they reproduce, then really the UK is not going to be a problem. So that kind of preconception that even if they get to the UK, everything will be fine because they won't survive our winters, I don't think that that's necessarily going to be true. So Tropilaelaps was confirmed in Russia in this area, again in 2024. And this graph is taken from a project, and the researchers looked at 50 colonies that were infested with Tropilaelaps, and the blue line shows the average of cells of brood in those colonies, and it uses, so this is the axis for the average number of cells of brood. So in January, there were about 200 cells of brood in a colony, and if it's sort of June, that goes up to about 1,400 cells of brood. And then here with the orange bars, you have the percentage of that brood that was infested with Tropilaelaps. And you'll see here that sort of February, March, April time, there was brood present because you've got that blue line, but there were no Tropilaelaps mites present. And these researchers, they checked the brood, and the Tropilaelaps mites had actually left the brood. And I spoke, so Victoria Sorka was one of the researchers that worked on this. I spoke to her at a conference, and I said, well, like, where did the mites go? Did they go on to the adult bees? And she said, no, we checked the adult bees as well. So they don't fully understand what's happening, but the theory is that the mites are leaving the brood for some reason, and that they're sort of secreting themselves in nooks and crannies within the hive, or potentially they're in that sort of hive debris at the bottom of the hive, but they weren't on the brood. So this preconception that they need brood all year round to survive, it's kind of evolving that that's perhaps not necessarily the case. So what do beekeepers in Asia do? So like I said, I've been to Thailand a few years on the trot now, and a few research projects. And the first time that I went there and worked with honeybee colonies, I was really quite shocked by how different Asian beekeeping is to Western beekeeping. So the first major difference is that the beekeepers in Thailand, so they don't use supers. They use these adapted Langstroth hives. You can see that the floor is attached to the brood box, so you can't actually separate that. And they have these, so this is a flap at the back, and they've got a flap at the front. And when you open up those flaps, you have ventilation panels front and back to be able to ventilate the hives. Now the hives are like this because the beekeepers practice migratory beekeeping. So in the same way that the Apis dorsata will migrate across the country to access different nectar sources, that's what the beekeepers are doing. And they'll shut up their hives, they'll shut the flaps, that will shut the entrance at the front, and they'll put the hives onto the back of a pickup truck, and off they go to the next sort of source of nectar. And the roofs also clip on with these metal hooks there. So the thing is, you don't want to have these transportable hives. You don't want supers on them. So what do you do to get honey? Well, they extract their honey from the brood box, which we sort of found quite strange. They also don't wait for their honey to be capped before they extract it. It's very watery honey, and they will evaporate off any excess moisture. So they have, they'll use sort of like these greenhouses, and they'll put the buckets of honey in these sort of hot sheds to evaporate the water off. So very different way of beekeeping to what we're used to. So the other thing is that the colonies are really small. So like I said, beekeepers want these transportable single boxes of bees, and you know they don't want to have to add supers. But the reason why the colonies are small is because of Tropilaelaps. So I've put two videos in here. Sorry, they're not the best videos. They were just sort of filmed on the fly while we were working over in Thailand. But this first video, so as part of the research we did, we bought 20 colonies, and they were delivered to the university, and were very standard. So they were 8 frames of bees, 7 frames of brood, nice colonies, good amount of bees in them. And this first video is probably probably about 4 or 5 days after we arrived. Now, this second video is approximately 3 weeks in to that work, and some of the colonies we didn't treat, so we just left them alone. They had no treatments for 3 weeks, and you can see how much smaller this colony is. So the reason why we're all sort of standing around looking at it is because we were concerned that our no treat colonies that they wouldn't survive until the end of the research, and that's wasn't what we were looking for. But you can see that there's a very patchy brood pattern. There's very few bees, and it's the Tropilaelaps mites that have done this to the colonies, because we didn't treat some of them for 3 weeks, and this is the kind of thing that we saw. So when we have beekeepers in Georgia and Iran and these areas where Tropilaelaps has just is an emerging threat, and they're reporting 2 to 3 months for colonies to die, and they're reporting sort of 80% colony losses. And initially we sort of took that with a pinch of salt, but when you don't treat, you don't do anything, and you see this sort of progression of the disease, it really is quite frightening. So what do the Asian beekeepers do to treat for Tropilaelaps? First time we were in Thailand, we witnessed a few concerning things. So one of the first things we were doing when we were out working with beekeepers, we chat to them a bit. We wanted to get to know about their beekeeping. And the very first beekeeper we worked with, I said to him, I was like, what do you use to treat for mites in your colonies? He went off to his shed and he came back with what you can see in the center there, which is this dog flumethrin-based flea treatment. And on here you can see he brought out this sprayer as well. And Dan and I were just sort of a bit bemused. And it got sort of slightly lost in translation, but he mixed it down, and we're not sure what he mixed it down with, but he was either spraying onto the combs or onto the bees, but he was certainly spraying it into the hives. And this is one of his treatments for Tropilaelaps, and we were just open mouthed. Like, I've seen some questionable kind of use of varroa treatments and varroa in my time, but at least these have actually been designed for use with bees. I've never seen anything like this dog treatment being used. I have pets myself. When I treat them for fleas, I'm always very conscious that
I'll call you when it's finished, right? Call you when it's finished.
I'm always very conscious that if I treat my pets for flea, that I don't sort of contaminate my hives with them. Certainly don't go out and work on my bees, because the fluvalinate and the flumethrin can kill honeybees. But the local beekeepers there, they've by a process of trial and error, they've developed these treatments themselves as a way to control Tropilaelaps. So on the right here, we've got a fluvalinate treatment from China, and these are just cardboard strips, and they are left on the floor of the hives for the bees to walk over them, and these are permanently in the colonies. So if you're in the UK, if you use, say, apple, for example, there are very clear instructions on how long you should leave the treatment in the colony for, and that's because you don't want the mites to develop a resistance to these treatments. So we became really quite concerned about chemical resistance because all of the hives we worked on, there were still healthy Tropilaelaps mites in these colonies, and they'd all been treated with things like this. So large parts of Asia, there's no control over the use of chemicals in honeybee colonies. We worked with beekeepers in the south of Thailand, and they were asking for advice because the bulk price for the honey that they were receiving was very low because of the contamination from these chemicals. And they were saying, well, what can we do? We have to treat for Tropilaelaps because our colonies will die. But if we do, then our honey's contaminated. And yeah, we couldn't sort of really give them much help. So 2024, we wanted to look at chemical resistance, and we kind of wanted to establish, is this really a problem, or is this just our perception? We wanted to see, we looked at the different miticides that are available, so the synthetic miticides, and we looked at amitraz, coumaphos, fluvalinate, and flumethrin. So the mites were resistant to coumaphos, fluvalinate, and flumethrin. Amitraz was 67% effective, which technically means that it worked, but I think you'll agree that 67% efficacy really isn't that sort of effective. If you still got sort of 30-odd% of the mites alive in your colony, that's not really going to do much for it. And it kind of shows that the mites in Asia are developing a resistance to amitraz as well. So these synthetic treatments really aren't going to work if they get to the UK, and we're planning on relying on those. So again, there's a QR code there if you want to read more about that one. So like I said, synthetic chemicals, they don't appear to be effective. There does seem to be quite high levels of resistance to it. So other researchers have looked at oxalic acid mixed with glycerine, and they've placed this on pads within the brood. There's been research into oxalic acid and thymol mixtures, where that's added to syrup and dribbled between the seams of the bees. Neither of these forms of treatment were that effective, so we tried, we had some pretty good results with oxalic acid sublimation. So we treated every 5 days for 25 days, so it was repeat treatment where we sublimated the hives. And we monitored the brood reinfestation, so how many mites were going back into the brood, and also phoretic mite numbers in the colony. and we had an 87% reduction in mite levels. The slight caveat to this, though, is that the colonies are a lot smaller than UK colonies. So by volume, the dose of oxalic acid we were using was probably higher than the same dose that a UK hive would, say it was a double brooded hive, the actual dose by volume would be smaller. And plus, the ambient temperature in Thailand is higher, so there is some evidence to suggest that ambient temperature affects the FC of oxalic acid when it's used to treat varroa. So while it is promising to see that something like oxalic acid does work, potentially, we do need to do more to look into this. So yeah, finally, to kind of wrap up here, I think we should be worried about Tropilaelaps. We should be aware of it. Like I said, they are more damaging than varroa, and I think what we've seen as they sort of progress through a colony and how that prognosis progresses, it is really quite frightening to see. The other thing is they're harder to spot in a colony. So if you're seeing parasitic mite syndrome in your colonies, it's easy to assume that it's caused by varroa, but if or when Tropilaelaps get to the UK, potentially they're going to be undetected for a while, because it's easy to assume that this is varroa damage that we're seeing and not Tropilaelaps damage. And they are coming this way. Like I said, the first time they were reported in Europe in 2024, and they're following much the same route that varroa did back in the 1980s. From talking to sort of other researchers that have worked with Tropilaelaps for much longer than I have, I sort of said, how long do you think we'll have in the UK potentially? And their best estimate is between 6 and 10 years. So, we're on a limited time here, and we do need to be aware, and we do need to proactively monitor our own colonies for them. The other thing is that there's very little research into Tropilaelaps. It's only recently that these discoveries have been made about how they can reproduce by parthenogenesis and how they overwinter in cold countries. It's like literally sort of 2-3 years worth of research, whereas with varroa, people have been looking into that for decades now. And treating and controlling these mites is difficult. Synthetic treatments, a lot of those synthetic treatments are still effective for varroa, but Tropilaelaps have already developed a resistance to them. So I'd like to thank you for listening to this. None of this work would have been possible without Bee Disease Insurance and Wax Chandler. Very grateful for the funding and their continued support. Chiang Mai University, they host us, they arrange for beehives for us, they give us lab space, and the team that come out to Thailand with me. Big thanks to Paul Davies, Dan Etheridge, Avril, and George Tong. This is very much a group piece of work that we do. I couldn't do it without them, so thank you to them. Yeah, and any questions from anybody?
All right, thanks very much, Maggie. You won't be able to hear the applause. Sorry about that. If you've got any questions, can you put them into the chat, please? Just a couple of points. Maggie will be speaking at the National Honey Show because the team are going back out to Thailand in about 3 weeks, aren't you?
Yeah, not long. We're packing up equipment tomorrow.
So she'll be able to update on what she's doing now. So get to National Honey Show and listen to Maggie. I've got a couple of other points here. You don't have to answer this if you don't want to, but why do you think the Asians are a bit behind in their research?
I think it's a bit of a different culture of beekeeping in Asia. So, and obviously, I don't sort of want to make generalizations, but a lot of what I've seen is very much sort of subsistence farmers. So you've got small holders, and they'll have, they'll grow crops, they'll have cattle, they'll have honeybees, and they've got this subsistence farming. And I think, because there isn't that more commercial honey production or queen production or package production. So nobody is a queen breeder. You know, I've never come across any sort of queen breeders or colony breeders in Thailand. They all just pick up swarms and reproduce their colonies like that. As I said, in terms of the honey sales, the issue that they have is that because of the Tropilaelaps mites, they have to continually prophylactically treat their colonies for Tropilaelaps, and that does cause a lot of contamination in the honey. So then when you've got the big bulk, they're beaten down on the price because the honey is contaminated, and they're sort of stuck in this cycle where there isn't an effective treatment for Tropilaelaps. Nobody kind of really wants to look into it and develop it because there isn't that beekeeping industry, and you know they can't sort of get out of that cycle, so they end up a bit trapped. So I think, and certainly it's only, you know, 2020 nobody was doing Tropilaelaps research. There was very limited. If you search scientific papers on Tropilaelaps before 2020, you're probably talking in the hundreds. The interest is growing now, but that's because it's come to Europe. It could potentially end up in North America. It could potentially end up in Australia, these beekeeping countries, and that's why people are suddenly taking note, because it will have a devastating effect on honeybee populations if it gets to, well, wherever it ends up next.
You mentioned the dog flea treatment. There were people in this country, honestly, and those of you who've been keeping bees long enough will be able to remember it. We're using sheep dip, you know, openly using sheep dip. So, you know, it's I see one head nodding there, so that they knew about it.
Yeah, I mean, we saw cattle mite treatments were being used over in Thailand, and I think it really, they've got to the point where it's just throw anything at the hives to see if they can get the mites under control. So yeah.
Yeah. There aren't any questions coming so far.
Yeah, we've got some, Roger. We've got some on. Yes, we have. Yeah, we've got a couple on YouTube. We've got a couple of chat here. I'll start with the YouTube ones actually. Maggie, has Tropilaelaps infestations in varroa-resistant colonies been investigated at all?
So it's not been investigated. I think the issue that I've seen in Thailand is because the progression of the infestation is so fast. So you are talking 2 months, 3 months for the colony to succumb to an infestation. I don't think the bees are getting that chance to develop a natural resistance. And also, everywhere I've seen, the bees are continually being treated, so that, you know, there isn't any treatment-free beekeeping going on. So I think the beekeeping has evolved enough there to be able to even try that when it's you know, when your livelihood's a bit more sort of dependent on it, and these, you know, these people are trying to sort of make a bit of extra money, that they'll sell the honey at the local markets, and that their default position is to just throw some chemicals at it, and they're not kind of worried about, you know, let's develop a natural resistance. They just want to harvest the honey and sell it.
Yeah, which might answer the next question as well, is are there any, or what resistant traits have been associated with lower infestation rates? So
yeah, so again, that's not necessarily something that we've really seen, and so university colonies and the colonies out with beekeepers that we visited around Thailand, it's very much the same story, where you do see varying levels of mites within the colonies, and some colonies will just succumb very quickly, but it is so much quicker than a varroa infestation to knock down the colonies that they really don't have a chance. And the interesting thing is, because if you've got a Tropilaelaps infestation, you tend to not have a varroa infestation, because so in that video, the mites seem to harass the varroa mites, and because they outcompete and they outbreed the varroa mites, the varroa don't seem to like reproducing in the same cells as the Tropilaelaps mites, so it actually reduces the varroa population, and then the Tropilaelaps sort of takes over. So yeah, it's when we were sort of doing filming and things like that, to actually get a varroa mite, it was like, oh great, we've got a varroa mite, so we can film it and we can put it with a Tropilaelaps mite for scale. But yeah, they're quite rare there because of the Tropilaelaps.
Frightening. Another one here about potential climate impact. Are infestations less intense in temperate regions like northern China or South Korea?
So no, I think certainly when I started with the bee unit 2014, I'd sort of come from being sort of small scale hobbyist beekeeper, and was trained up to be a bee inspector. And in 2014, the training for Tropilaelaps was, you need to monitor for this mite, but don't worry too much, because even if it gets to the UK, it won't become established, because the mites need brood all year round to be able to survive. And we all just went, okay, fair enough. But then so South Korea, China, all of these countries, and now if you look at areas in Russia where Tropilaelaps have been established, considerably harsher climates than us, and temperate climates. And like I said, nobody kind of fully understands how they're surviving, but they are surviving and reproducing in these areas. So while they are a tropical mite, they do seem to be able to kind of evolve and adapt quite quickly to changing climates. And if they end up in a new area, they do seem to be able to do quite well in that area.
Okay, turning some of the questions on the chat here. This one's a bit above my head, but I'm learning a new word. Tell us, please, what you know about the possibility of a tick being in diapause at a certain stage of its development.
Yeah. So there's one piece of research from Russia which looked at egg diapause. So diapause is, I kind of think of it like a sort of state of suspended development. So if the environmental conditions aren't sort of perfect for insects and some mites, they can get into this state of diapause, and it's sort of like suspended animation, and they can stay in that state for a while until the conditions favorable. So like I said, there is a piece of Russian research which looked at egg diapause, which showed that eggs were viable. I think it was 8 weeks later. I think they chilled them down to sort of 0 degrees or 2 degrees, and the eggs would then still hatch out. We're interested in adult diapause. So we had part of our work, where we did the survival study, and we had the mites on the different matrices. We had mites left over from that, and some mites actually sort of accidentally ended up in the fridge. We kind of realized that, and at the end of the project, we went to kind of clean everything out and tidy up. And I thought, oh, all right, I'll tidy that up, and assumed that the mites would be dead, because I think they'd been in the fridge or the freezer for about 5 days. Put it on the side. The mites defrosted, and they were still alive. So although not scientific, we were all astonished, because there was this pot of mites that had been chilled down, no food, no protection, anything like that. And they were certainly very much alive and kicking when they were brought back up to room temperature. So that is something that we want to go back and look at, but very, very limited work on it at the moment. Tropilaelaps are Laelapidae mites. Other Laelapidae mites can enter diapause. So there is, they have got for doing this.
Right, frightening. Question here: was Formic Pro tried as a treatment?
Yes, so one of the pieces of work that Samuel Ramsey from, he used to be USDA, I think he's moved university now, but if you sort of Google Dr. Sammy or Samuel Ramsey, so he did some formic acid work, and I believe he used Formic Pro. So his results were that it was effective at kind of knocking back Tropilaelaps and reducing the mite numbers. But when you read the paper, he also observed that the mite numbers built up very quickly again afterwards. So, and in the graphs that he's kind of produced for this paper, there is that big knockdown in mite numbers, but then it climbs quite quickly again the other side. And I think he tried that in conjunction with, so in the United States, they'll do heat shocks, where I think they'll heat the colony up to 42 degrees. That might be wrong, but it gives that sort of heat shock to the colony. It's quite effective for controlling varroa mites. And they tried that with Tropilaelaps, and again, they saw a good initial knockdown with mites and a reduction in mite numbers. but again, the numbers back up very quickly. Beekeepers in Thailand and that they will use formic acid, but it's a bit more hit and miss. They'll mix up their own sort of concoctions and they'll put it on cotton buds and they'll, you know, introduce it to the hive in various sort of different ways. So it's kind of hard to know what sort of dosage is effective and for how long to use it. But it does seem to work, but it's a bit of a grey area.
Okay. Couple of questions here about whether brood breaks might have any impact or be effective.
Yes, so Rogan Tokay, so he is Auburn University. I think he's just published some work with so oxalic acid and with brood breaks. I think the brood breaks were, I can't remember off the top of my head, the sort of percentage of mite knockdown. So he did have good results from brood breaks. I think the only sort of slight concern is that the length of the brood break that you need to be able to manage is difficult in kind of a beekeeping situation. Certainly, like the more commercial beekeeping situations, beekeepers would struggle to do that. So, and I think whatever treatments end up kind of being used, it needs to be something that's actually practical to do on a large scale. As you know, it's all very well hobbyists doing it, but the commercial beekeepers need to be able to do it. So brood breaks do work, but it's one thing doing it in a sort of you know, lab field environment where you've got the time and you've got the resources. It's then scaling that up to be able to do it on a sort of wider scale with commercial beekeepers and, you know, even sort of novice beekeepers. It can be quite a hard thing to do and sort of manage a brood break without the colony getting, you know, sort of potentially going queenless and things.
Yeah. Okay, good. One last one, I think here. Is there a chance that Apis mellifera will learn to deal with Tropilaelaps as they have learned to deal with varroa?
I think I'd like to hope that there is. I think, you know, I've not seen evidence of that, but that's purely just because of where Asian beekeeping is and where, you know, where Tropilaelaps are. And I think the more we can sort of research it and the more we can better understand it, they are a very different mite to varroa. So it's easy to kind of make the assumption that they're both sort of honeybee mites. Like I said, you know, varroa mites, they can't reproduce parthenogenesis, and, you know, varroa mites, they can't mate outside of the cells, and there's this whole different way that the mites behave that isn't fully understood, and that might be, you know, beneficial, and it might make it easier to control them, or it might make it more difficult to control them, but we just don't know that at the moment. And I think that's the thing. And every time I go and work with them, you know, we're always kind of shocked by you know, what we see and what we do. And I think, you know, we were a bit blasé the first time we went and worked with them, and yeah, it kind of came home quietly to roost that actually this is something that, you know, we need to take seriously. And so I think don't underestimate them. I'd hope that our bees would be able to develop some form of resistance to it, but yeah, but I wouldn't sort of be complacent, if that makes sense.
No, lots more to learn. Okay, thank you, Maggie. Roger, over to you to.
Right, yeah, Maggie, we really have to be serious about this, and it's good that you've mentioned what you have towards the end. Somebody's really got to lead this, and we tried to do something for the inquiry, the AFA inquiry. We tried to get people to concentrate on Tropilaelaps. I'm hoping a lot of beekeepers have responded. I'm old enough to remember when a varroa came, and I must be forgiven for not having a 30-year-old memory, but as I remember it, varroa was first discovered in April 1992, so it obviously been here over winter. I can't quite remember the figures, but as I seem to remember, that year there were 200 to 300 colonies that were discovered with varroa as well. So it had just got out of control very, very quickly, very quickly. Those figures would need a checking. My concern is that a lot of beekeepers can't actually see queens, and varroa mites and Tropilaelaps are a little bit smaller. And I think if they see anything, they will just see it as a pollen mite or something in the hive. So I think what we've got to try and do is, if we possibly can, make sure it doesn't come here in the first place. And one of the issues that both you and I have sort of discussed on email a little bit is charities bringing or encouraging beekeepers to buy queens from areas where it's thought that Tropilaelaps is in existence. So I think we've got to do quite a lot of work and highlight these things. And so somebody somewhere along the line's got to do it.
Yeah, I mean, I think prevention is definitely better than cure in this instance. So when the initial idea came about to work with Tropilaelaps, so the initial idea was to develop screening techniques for the National Bee Unit. So how could we monitor for Tropilaelaps more effectively? And so there's myself and Dan and I have worked together for 10 years, and Paul and George are seasonal bee inspectors with the bee. And we went out to Thailand, and it was like, great, let's go and look at some Tropilaelaps mites. And we were sort of quite, I don't know, probably quite cocky in that. And went to Chiang Mai University, and we had hives there, and we knew that there were Tropilaelaps mites in those hives. And between the four of us, probably 100 years of beekeeping experience. Dan's been a commercial beekeeper, Paul's a commercial beekeeper. I've done, a slightly smaller scale than the guys, but I've done lots of research, and we're like, right, let's go and find some mites. And I think 3-4 hours later, we were still in the apiary and we hadn't found any mites. And it was really quite frightening because, we work with bees all day, every day, and we couldn't see the mites in the hive. And we sat there and we went, if we can't see them, how the hell is anybody else in the UK going to be able to see them? And then the script techniques that we were using didn't work, and we couldn't find the mites. And we've developed new ones now, but that was a real wake-up call for just how difficult they are to work with. And I'm always conscious that I sound a bit sort of melodramatic, but I can't emphasize enough just how hard they are to see in a colony. And I've worked with them 2 years on the trot now. I know I go back in March, we will be struggling to sort of get our eye in and to see them and to sort of kind of get to sort of work with them again in the colonies. And if you've never seen it, you just don't know what you're looking for. And it took us a couple of days of, we know colonies mites in, right, let's go and find them, and it took us a while to be able to spot them reliably in the colonies.
Well, just a couple of points for me left. You showed just one bee on a frame, and that clearly had chronic bee paralysis virus to me. That presumably is a lot higher level than it is with varroa because of the extra infection in the pupa. Is that right?
Yeah, and I think so I sort of briefly touched on it, but it's not just CBPV, it's all of the viruses that you would get in a honeybee colony are vectored by Tropilaelaps. And when you think of things like so we don't necessarily have much sort of black queen cell virus or Israeli acute paralysis virus. You don't hear of it that much in the UK, but when you talk to beekeepers in Taiwan and Korea and Thailand and places like that, they have lots of issues with those viruses. And when they look at it, so like I said, it's different strains of the same viruses that kind of we already kind of have in the UK, deformed wing virus, things like that. It's those different strains that, like with COVID-19, if the population doesn't have some level of natural immunity to it, then when that virus hits for the first time, it will just wipe out a population. So that's another, it's lots of different factors that all kind of play into the way that Tropilaelaps are such a threat to the colonies.
And do you still see parasitic mite syndrome with Tropilaelaps?
Yes, so that's when I showed the video with the 2 hives. I mean, I've never seen parasitic mite syndrome progress so quickly. But like I said, we had our 20 hives on day 1, and they you know, we wanted hives with mites in because we had this work to do. And when we turned up, and the beekeeper that we worked with supplied us with these lovely hives, and we were like, oh great, like thank you very much. But when we got back to the lab, we go, oh bloody hell, we've got these hives that are almost too perfect and too nice, and we're not going to have mites to work with. But these very healthy hives, the Tropilaelaps infestation so quickly that by week sort of 2, week 3, oh okay, well it's not a problem. We were concerned we wouldn't have enough mites to work with. It wasn't a problem. We were worried whether some of the colonies which we hadn't treated would actually survive until the end of the month that we were going to be there. And it's a completely different way of looking at it. The sort of go down to the apiary and open up the hive, and you could just see it deteriorate over 2 days later, 3 days later, a week later, and that's what seeing that sort of firsthand is really quite shocking.
Okay, Maggie, thanks very much. Is anything else coming, Steve?
One more question on YouTube. Does pin tests do what is it on this? On the current point, does the selection on pin test for its hygiene do well or better against this new parasite?
So I've not sort of heard of any work that kind of looks at that sort of hygiene behavior of bees. I would kind of assume that any bees that have those hygienic tendencies and that pull out the dead brood would fare better with Tropilaelaps. Like I said, with any population, and you'll probably see this in your own hives, you'll have some hives that are very hygienic and very productive, and might be a bit sort of snarky and a bit peppery, and then you have the very docile hives that perhaps aren't terribly productive, and you get this range of sort of traits and different qualities that the different hives have. So some will just naturally be more hygienic and more predisposed to groom themselves to remove Tropilaelaps mites and things like that. So I think things like freeze drying the brood and pin tests and things like that is a good way to check for hygienic behavior, and certainly it's going to help with Tropilaelaps mites because hopefully the bees will pull out any dead brood, will groom themselves, and will sort of clean the mites out of the colony.
Okay. And one last comment. Apparently, JF says Kyra Wagner from UBO is investigating hygienic resistance to Tropilaelaps in South Korea.
Oh, brilliant. Okay, that's good to know.
Yeah.
Okay, well, thanks very much. We'll wrap it up now. Thanks very much indeed, Maggie. And for the listeners tonight, and also on the recording, please get information links to your local beekeeping association so that as many beekeepers as possible are aware of it. So it just remains for you. Thank you very much on behalf of the listeners, Maggie. Thank you all very much.